In this video, our objective is to investigate osmosis in plant cells. What is osmosis? Osmosis is a special case of diffusion. It is the movement of water from a dilute to a more concentrated solution through a semi-permeable membrane, which allows water to pass through. Plant tissue reacts strongly to the concentration of external solution that you can use as an osmometer or a way of measuring osmosis. Potato is commonly used as the experimental plant tissue. Sweet potato and beetroot can be used as well. In our experiment, we will be using potato. Now let's go through the procedure. First of all, we cut the potato into cylinders, rectangular chips or small discs, all of the same surface area in order to compare our results easily. As a safety measure, take care when using sharp instruments. Then weigh the mass of the potato chips using a balance. These potato chips are made up of many plant cells, and within each plant cell, there is a certain proportion of water and dissolved substances like sugars. Now, we will take the potato chips and place them into two beakers, each containing a different sugar concentration. In the first beaker, the solution has high sugar concentration, meaning that the concentration of water in the solution is low, even lower than the concentration of water inside the potato chips. Due to osmosis, water at high concentration inside the cells of the potato will move out of the potato through the semi-permeable cell membrane to a lower concentration of water in the solution that the potato is surrounded by. As a result, each cell of this potato chip will lose turgidity and the overall potato chip will shrink to lose mass. While in the second tube, the solution is diluted, meaning it contains high water concentration. So water particles will move by osmosis towards the lower concentration of water inside the potato chip. As a result, the potato chip plant cells fill up with water, causing them to swell and gain mass. Now we will wait for 30 minutes, then we will reweigh the potato chips and record our data. Dry the potato chips thoroughly using a paper towel so that you remove any excess liquid from the plant tissue, because it might affect our results. So now, what to do with this data? We have the independent variable, or the variable that we are changing, which is the concentration of the sugar solution, where we placed our potato chips, and we have the dependent variable, which we measure, where we measure the percentage change in mass of the potato chip. And we have the control variables, which should be kept constant for both potato chips to have accurate results, which are the surface area and duration of the potato in the solution. To answer the question, we generate results in the form of table. It includes the varying concentration of sugar in solution, the starting mass of the potato chip, the mass of the potato chip after 30 minutes, the change in mass, which is the mass after 30 minutes subtracted by the starting mass. And once you calculate the change in mass, you divide it by the starting mass and then multiply it by 100 to get the percentage change in mass. So when the potato chip is placed in pure distilled water, which is of 0% sugar concentration, the percentage change in mass was 100 after 30 minutes. At 10% sugar concentration, the change in mass was 68. At 30% sugar concentration, The change in mass was -6, and so on. In conclusion, as the concentration of sugar solution increases, the percentage change in mass decreases. And if we plot these results on a graph, we will be able to indicate where the isotonic point is. This is the point where the potato will neither gain or lose any mass.